Hello everyone, and thank you for attending today's webinar, Molecular Pixelation, 3D Spatial Proteomics of the Cell Surface. I'm Tristan Free, Senior Editor of BioTechniques, and I'll be your host for today's event. Before I introduce you to our speakers, I'd like to quickly cover a few housekeeping items. You've joined the presentation and will be listening via your computer's audio by default. If you would prefer to connect to a different speaker, you can adjust your audio settings by clicking the arrow next to the microphone icon at the bottom of the Zoom window. You'll have the opportunity to submit questions to today's presenters using the Q&A tab at the bottom of the Zoom window. You may send in your questions at any time during the presentation, and we'll collect these and address them during the Q&A session at the end of today's webinar. Please let us know your thoughts on today's webinar by tweeting us at MyBioTechniques using the hashtag BTNWebinars. So we are delighted to have Petter Brodin and Jessica Nordland presenting for us today. Petter is a paediatrician and professor of paediatric immunology at Imperial College London and the Karolinska Institute in Sweden. His lab in the Karolinska Institute develops and applies novel experimental and computational methods to describe human immune system variation with a particular interest in the immune system of children, its development in early life, and its role in health and disease during childhood. Jessica is an associate professor at the Science for Life Laboratory in Uppsala University, where she serves as director of the Swedish National Genomics Infrastructure and leads a research group. Her research focuses on integrating high-throughput ex vivo drug screens and omics data with machine learning to pinpoint biomarkers for the refinement of molecular diagnosis and to investigate innovative approaches for the treatment of acute childhood leukemia. Thank you for joining us today, Petter and Jessica. I'll now hand over to you, Petter, to start today's presentation.
Thanks very much, Tristan. It's great to be here and share our excitement with this new technology, molecular pixelation, which is adding a new dimension to our studies of immune cells in our context of human immunology. So if you go to the next slide, please, Jessica. So the immune system that we study is a distributed system of specialized cells, each performing unique different tasks, which gives the system as a whole an ability to perform more complex tasks. But to understand this system, we really need to study all these different cells in combination, but also in great detail at single cell resolution to understand what determines activation of a particular cell type, what distinguishes a cell as it moves along differentiation trees, etc., etc. And here is where single cell studies really come into play. Next slide, please. And traditionally, the most important technology for us as immunologists have been cytometry methods, such as fluorescence-based cytometry, mass cytometry, and more recently, sequencing-based methods dependent upon oligonucleotide tagged antibodies and then sequencing. So in fluorescence-based flow cytometry, cells are stained, they're proteins, targeted by antibodies labeled with fluorescent proteins, and then studied one by one using a fluorescence-based cytometer. And that allows you to measure 10 to 15, up to maybe 30 parameters per individual cell for millions of cells. And this has allowed us to distinguish the different populations of cells that constitute immune systems and their differentiation and activation states. Next slide. But all of this is based on protein abundance. And protein abundance is measured as a intensity of that fluorescent signal in flow cytometry. And we've studied, as I said, 30 or 40 or so parameters per individual cell with respect to abundance. What molecular pixelation allows us to do, if you take the next slide, is to add additional dimensions to this type of single cell study, because it allows us not only to look at the abundance of a protein, but also the distribution of a particular protein of interest in a given cell, and finally, colocalization patterns across a number of different proteins. So these are brand new dimensionalities in our data of single cells, which we have never been able to study at this level of detail before. Next slide. So there have been methods to do similar things previously based on imaging of cells, But those are limited in throughput and in dimensionality. So the number of proteins that we can study using imaging methodologies is limited. And what molecular pixelation allows us to do is to scale this up, in theory, to hundreds of proteins in individual cells for hundreds, if not thousands of cells based on sequencing. Next slide. So to give you an example of why protein localization in immune cells is important, I wanted to remind you of studies which sort of was really sort of at the forefront of immunology 20 years ago or so, which was focused on the immunological synapse, in this case between a T cell and a antigen presenting cell. And what these types of studies were showing, primarily using single cell and single molecular imaging tools, and was that protein colocalization around this synapse was a really important way to tune both the strength of a signal sensed by the T cell, its activation, its differentiation signals, but also the qualitative nature of these cells, these signals. And so protein localization, as you see in this image of an immunological synapse, really determines function of the cell. And this is a dimension that we since have sort of forgotten about and not studied it at this level of detail. Next slide. So molecular pixelation allows for this to be done now. And the way that this works, I'll briefly talk you through this. And as you can see at the bottom, there is a paper describing this in detail, which is soon to be published. But basically, cells are stained using antibodies tagged with an oligo tag, each unique for the antibody in question. So cells are stained after fixation, and that allows for targeting of the proteins of interest, up to 100 or so proteins currently. Then there are 2 rounds of pixelation, which connects these antibodies to a spatial pixel A and pixel B, through these gap-fill ligation methods that allow us to expand a barcode, basically. And so in the end, each antibody will have 4 barcodes: 1 UMI, unique molecular identifier, a protein barcode, which tells you which protein is targeted by the antibody, and 2 pixels, pixel A and pixel B, which allow you to form a localization of the antibody signal across a cell. Next slide. So in sort of data analysis terms, what this allows you to do is to study not only the binding intensity amount of protein on a given cell, but also the way that those proteins form neighborhoods in a cell, and finally, across the entire cell, how those neighborhoods connect to form the spatial distribution of proteins on a cell. Next one. And in real terms, the way that this data is processed is through this form of nearest neighbor graph, which represents the data from the sequencing result. Next one. And so what do we do with this type of data? And the focus of this webinar, other than introducing the technology, will be to give you a few snippets of where we are beginning to go with this technology, both within my lab and then after me in Jessica's lab. And what the first thing that everyone who studies the immune system would want to know is, what cells am I capturing here? And what this result shows here is a PBMC data set, so peripheral blood mononuclear cells. And as one would expect, using the abundance of proteins, it's relatively easy and straightforward to identify the different populations that we are normally recognize in blood. Next one. But what's intriguing is that looking at the spatial distribution of these cells, in certain contexts, for example, when a cell is activated or when a cell is moving, migrating, such as in this example here, showing T cells migrating on a glass slide coated with ICAM1, cell distribution, or the protein distribution in a cell, is altered. And what this example, if you go to the next slide, shows is how specific proteins are localized to the uropod fraction, or the leading edge fraction of an individual T cell immobilized in this way, which really begins to it illustrates this new dimension of data that we currently have not considered much in our single cell experiments of immune cells. Next one. Another really intriguing example comes from this data set generated by the Pixelgen team, where cells stained were found to have a graph which is shown on the left, which indicated two separate entities co-localized in this graph. And when looking at specific markers, it was evident that the left part of this graph represented a platelet and the right one a monocyte, which then illustrates a cell-cell conjugate or interacting entities, which allows us to now begin to study at real level of detail how cell-cell interactions might actually occur using cells in suspension. Next one. And then from my own lab, what we're currently using this tool, one example of what we're using it for, is to study this really intricate process in which a naive CD4 T cell receives a signal, an antigen that it's and then decides whether to become a Th1, Th2, Th17, or a regulatory T cell, all with very different properties and functional response mechanisms. But the determinants of what that entails, why a cell chooses a particular lineage over another one, and what actually distinguishes these different cells and their functional properties is not very well defined. We know some of the transcription factors which regulate these decision-making, such as the ones illustrated in the middle of these cells here. But what really functionally differs between these cells is not fully established. So what we have done in our lab is we have cultured cells in vitro from healthy human donors and polarized them in one of these directions, and then studied them by molecular pixelation to try to understand what actually differs between these cells. Next one. And one really intriguing example comes from this analysis here, which shows polarity score. And polarity score from molecular pixelation data is a way to quantify the spatial distribution, whether a particular protein, in this case CD82, is either polarized or evenly distributed across the cellular surface. And what we show here is that for CD82, this protein shows a marked difference in its distribution pattern, with much more polarized distribution in the Th17 type cells as compared to the uninstructed Th0 cell. And what's intriguing about this is that CD82 has been shown previously to be increased in abundance upon cellular activation, but its distribution changes have not previously been well studied and has not been shown to differ with respect to Th17 cells. And we are really intrigued by this, following this up with signaling studies to try to understand how this might impact the signaling downstream of the T cell receptor in these cells. Next slide. A completely different readout from the exact same data is colocalization scores, which basically allows us to quantify the way that different proteins are either colocalized or repulsed basically from each other within individual cells. And that's illustrated for a few markers comparing Th1 versus Th0 type cells on the left, Th2 or Th0 on the right, showing some very similar things, such as CD48 being very similar, while other markers differ between the two, suggesting that the distribution of protein-protein interactions or protein-protein pairs actually mediates some of these differences between these cell types, which is likely to impact signaling of the cells, activation states, etc. So next slide. And so to summarize my part of this talk, molecular pixelation adds a whole new dimension to our single-cell protein studies by providing a protein-protein interaction analysis, as well as a spatial distribution of many proteins in individual cells through sequencing. This would offer brand new biological insights into any system involving single cells and protein dynamics. In my case, we're particularly intrigued in how this could explain differences between seemingly similar types of immune cells, such as the CD4 T cells that I showed you here, but also how this might actually explain good or bad responses to therapies, vaccines, etc., etc. And we're very happy for the collaboration with Pixelgen, as well as the support from Knut and Alice Wallenberg Foundation, who's offered us a proof of concept grant to continue this work. And with this, I'm really happy to hand over to Jessica, who will tell you a bit more about a completely different application of molecular pixelation in leukemia.
Thank you, Petter. So now I'm going to take over here and talk a little bit about what we're doing in leukemia cells, specifically in B-cell leukemia, looking at mapping drug responses ex vivo. So just a little background about acute lymphoblastic leukemia. It is the most common pediatric cancer. It arises from the immature lymphoblast cells, so either B or T cells. I'm going to be mainly speaking about B cell ALL today. It's an acute disease and it requires immediate treatment. And currently the treatment protocols last about 2.5 years of intensive combined chemotherapy. And the implementation of protocol-based treatment regimes for ALL is probably one of the greatest cancer success stories. When these treatments were started in the 70s, very few patients survived their diagnosis, but today nearly 90% of patients survive. However, as the first childhood cancer survivors are now reaching midlife, we're realizing that the survival actually does come at a cost. Sweden has amazing health registries that can offer comprehensive longitudinal data. So this has allowed the follow-up of the nearly 10,000 childhood cancer survivors in Sweden today. And the results are quite concerning. About 7 out of 10 of them experience at least one major complication from the treatment they received as children, and about over half of them are serious or life-threatening. There's an overrepresentation of diseases such as diabetes, cardiovascular diseases, infertility, secondary malignancies, accumulating in about a 40 times higher lifetime mortality rate than their age-matched non-cancer survivors. So what we're working on now is to try to better understand which patients need the intensive combined chemotherapy. So which patients need more treatment, and which patients do not need as much treatment. And current genetic risk classification has come a very long ways, but it doesn't tell us everything. So in some of the genetically defined subgroups, we can see that even though there are good prognosis groups, we do see just in pure numbers the most of the relapses. So we need better risk stratifying markers, and we need to do better functional characterization. So something that has been ongoing in the Nordic countries for many years is something called functional drug screening. So this is an assay where you take the blasts, so the leukemic cells from the bone marrow, you take them out of the body and you put them in an ex vivo drug screen. And then you measure the response to, in this case, in the study, the 8 different drugs that are given during the treatment, and trying to measure whether a patient will be resistant or sensitive in vitro, and then relate those findings to in vivo. And these studies have been quite successful. I mean, we've really shown that what we can see here is that patients who are sensitive to drugs in this ex vivo screen tend to do better. So here in this Kaplan-Meier curve on the left-hand side, you see the blue line represents the patients who are the most sensitive to the drugs in this ex vivo drug screen. Then the purple line in the middle would be those who are intermediate, and then the red line being those who were the most resistant. So what we can clearly see here, and this is just one example of one drug, which is cytarabine, which is one of the what we call the backbone drugs of the treatment, that if the patients are resistant ex vivo, they tend to do poorer in their outcome. And in this large study, is a Nordic multi-center study with about 900 patients, what we could see is that it seems that there is something in the cells already before they start getting treatment that we might call it a cell state. I'm not really sure if I'm ready to say that yet, but some sort of a priming of the cells that seems like that they're going to push them towards resistance or sensitivity. And what we are now looking at is trying to figure out, so we can do these ex vivo drug screen, but can the phenotypes of the cells that actually survive these assays tell us something about their sensitivity? Can they also tell us if there's a way of their resistance, or if can they tell us something about how we can target them? And so that's exactly what we're trying to do here. And so in this proof of concept study, we wanted to do single cell RNA sequencing in the cells after they've been treated with drugs. And if you can imagine the situation that we're in, we're bombarding these cells with a lot of cytotoxic drugs. And so we're having a lot of dead cells in the sample that we're working with. So it's a very messy sample. And so to start with, we started looking at 3 single cell RNA sequencing methods. I've limited here, and I'm just going to kind of breeze through this since this is published data. But what we did was we used these 3 methods and looked at the cells that were exposed to 5 different drugs and DMSO as control. And we can see that all of these methods worked pretty well. And what popped out of the study was that the cells that were treated with a drug called fludarabine, they tended to look very different than the other cells. And when we see this donut-like shape here in the analysis, that makes us always think about, okay, this looks like cell cycle, and indeed it is. And we can see that these cells here in the lower left-hand corner that pop out there, it seems like they're kind of stuck in, the fludarabine-treated cells are stuck in S phase. And this makes perfect sense. Fludarabine is a purine analog, it's an antimetabolite, and it inhibits DNA synthesis. And in these cells, we see that we do see an increase in genes in the p53 pathway. So this all kind of makes sense for what we're expecting. And so just as a summary, this first part of the study is that we can see that the ex vivo drug screens they're giving us a very good amount of data, maybe potentially helping us stratify patients into groups that are more likely to benefit from specific treatments. And using the single cell data, this has really provided an additional layer of information. But one of the things that we noted when we did the analysis is that the fludarabine treated cells, we saw a little bit, or a lot of genes that were encode cell surface proteins that were upregulated. And so that's why we decided to go ahead and test this molecular pixelation assay. And now Petter's gone through this in detail, so I'm not going to say so much about it. But essentially, in our hands, what we do is we isolate the cells, we fix them with PFA, we incubate them with the oligo conjugates, and then, as Petter explained, you do these 2 rounds of molecular pixelation to kind of identify the neighborhoods, bringing those antibodies that were in close proximity to each other into a DNA tag, which we can then turn into a sequencing library, sequence and analyze. And by doing this, we get the abundance, the spatial distribution, and the colocalization data. And so just since I don't have so much time, I'm just going to show one vignette over one of the experiments we've done. And in this experiment, we've taken B leukemia cells, so pre-B cells, and we've treated them with either DMSO as control or fludarabine for 72 hours. And then after 72 hours, we've collected them and processed them for molecular pixelation. And so first, looking at the protein abundance, what we could see here in this volcano plot, anything going towards the right then would be upregulated in abundance in the fludarabine treated cells compared to downregulated. So CD71 is downregulated, and for example, CD40 seems like it is upregulated after fludarabine treatment. And since we had the single cell RNA-seq data, what we did is we made a pseudo bulk experiment here, and we looked at the relative RNA-seq expression levels for these genes and the protein abundance. And I was actually a bit surprised when we saw this, that we did see generally a rather good correlation between those genes that we saw that were upregulated in the single cell RNA-seq data. We also saw that they were, many of them were upregulated in the MPX data. So moving on to colocalization, and this is where we see which proteins are more often close to each other or segregated from each other. And we can see that there is a lot of what we can modulation of the cell surface after fludarabine expression. And then when we looked at polarity, and interesting, it seemed like after the treatment, we only saw upregulations in polarity. And among the top proteins that changed in polarity was actually also CD82 in our study. And so what this showing here is just a example of representations of any given DMSO or a single DMSO treated cell. You can see that there's some CD82 expression, but in the fludarabine treated cell, you can see that there's much higher expression of CD82 on that side. And then next, to see how this looked like, we went in with another method, so just doing very standard immunofluorescence assays. We looked at the DMSO cells, which are here on the left, and then the fludarabine treated cells on the right, and then they're colored with DAPI in blue, and then the magenta is the anti-CD82, and you can see very clearly that there is upregulation of CD82 there, and in some cells we do actually see the polarization. And one of the interesting things that we also noted here, and this is something that we have noted in other studies, was that the fludarabine treated cells are actually a little bit larger, and this is something that we could also look at in the MPX data, where we could using the number of tags on the cell surface kind of get an estimation for how big the cells were. And we can see that the fludarabine treated cells do also in the MPX data look like they are bigger than the DMSO treated cells. CD82 is upregulated in AML and CML and CLL, but there hasn't been so many, so there's not so much information in the literature about ALL, and it is also specifically in AML known to be upregulated and associated with chemoresistance in the bone marrow microenvironment. So we think this is very interesting, and it's something that we are going to take a closer look at here moving forward. So these pictures here just show the protein neighborhoods of two representative cells, so one DMSO cell and one fludarabine cell. And each one of these little, I can say little dots is a neighborhood of proteins, and then they're colored based on how much CD82 expression. So you can see here on the top of the fludarabine treated cell, where we can see a high level of CD82 expression. So in summary, we found that the fludarabine seems to induce differential expression in the surviving cells and modulates the cell surface proteins. And we've seen this both now on the single cell RNA-seq level and also with the molecular pixelation data. The assay works very well in our hands, and it's really given us a nice opportunity to kind of look at a problem maybe with a new set of glasses put on, because we didn't before have the possibility to kind of, look at so many proteins at the same time, and also look at, where they are in relation to each other, and not only just looking at the abundance, because if we would have looked at the abundance alone, we probably wouldn't have found this observation. Then we have a lot of questions that we're not answering now. So is the upregulation of CD82 specific to fludarabine? Is it specific to this one type of leukemia that we're looking in, or is it a general effect after exposure to cytotoxic drugs? And is it actually doing something that we should be interested in? So with that, I would like to thank Pixelgen for the opportunity to be a beta tester, and also thank everybody for listening and the opportunity to come here and present our results. Also the patients and the funders who have provided funding for the study.
Fantastic. Well, thank you so much for your, thank you so much for your presentations, Petter and Jessica. If you do have any questions for our experts, then please continue to submit them using the Q&A button at the bottom of your screen. We've already received a number of your questions, so we'll start addressing some of these now live on air. So first off, for you, Petter, why would protein polarization and protein-protein interactions offer additional information rather than protein abundance alone?
Right. So this goes back to this issue I briefly touched upon in the beginning, which has to do with the localization of proteins in a given cell actually modulates signals and modulates motility and movement of cells. And so by capturing this dimension, it makes us able to detect changes associated with these features that would not be detectable from abundance alone. We've seen in our analysis of molecular pixelation data that some proteins do correlate between abundance and, let's say, polarization, for example, but other proteins show different patterns. So they might not change in abundance, but increase in polarization with a given stimulation, for example. So that really these are orthogonal measurements that provide additional information.
Fantastic. And then Jessica, in terms of what is it that makes MPX suitable for hematology studies? What's particularly good about it for these type of studies?
I mean, yeah, so when we're working with these type of cells in suspension, it is this, according to me, is really one of the only, spatial proteomics assays that we have available to us, because our cells are not in a block and they're not in a tissue. And here we're actually able to get the 3D representation of the entire spherical cell. And yeah.
Okay, fantastic. And then again for Petter, what are some of the key applications that you see for these methods in the future?
Oh, I think there are numerous, and it's all about the imagination of different researchers. But anyone who is interested in either in a particular protein and looking at what binding partners this protein might have, or individuals who are studying cells moving in a particular way upon activation, etc., what determines the mobility of these cells, then molecular pixelation would really offer a high throughput and high dimensional assay to test these things. For us, we think of cell activation events, cell-cell interactions in the context of an immune response, etc. So I think there are numerous applications in immunology, in cancer, as Jessica has shown, but also in basic cell biology and so on. So.
And then for Jessica, what is the the optimal sequencing depth required per cell when you're using it for your applications?
Yeah, that's a great question. So we have to date done 120,000 reads per cell, which is quite deep. We've sequenced some cells to up to 200,000 reads per cell. And I want to say we're working on doing downsampling experiments. So I don't think that you need that high depth in order to get a very good quality library. But right now the recommendation is 120,000 reads per cell.
Okay, fantastic. And then following on from that, which sequencing setup or instrument do you recommend for this specific assay?
Yeah, so to my knowledge, Illumina NextSeq, NovaSeq 6000 and NovaSeq X Plus have all been used to generate libraries successfully. Right now, since the kit comes in a pack of 8, we are typically running 8 samples or 8 analyses, 1,000 cells per sample on 1 lane NovaSeq X Plus. Okay. It kind of gives the sweet spot with the right amount of sequencing for that setup.
Brilliant. And then Petter, for you, what are some of the next steps? How would you validate your MPX data?
Yeah, so when looking at findings such as the CD82 one that we had in our TH17 cells, the obvious thing is to do just like Jessica showed for her cancer studies, looking at imaging data when you know a particular protein that you're interested in. The problem in the past has been that we've started with imaging, but obviously we can only image, 1 or maybe a handful of proteins routinely. And so starting with the molecular pixelation, which gives you this large panel at high throughput, gives you a sense of what proteins might change in the most interesting ways. And then you can dig deeper into those using imaging as, I suppose, the gold standard method for a single protein and its distribution.
Excellent. And then, Jessica, based on your experience, how does cell viability impact the success of these studies?
Also a very good question. So as I said, we kind of are bombarding ourselves with cytotoxic drugs. So we have a lot of cell death. And we've managed to, just kind of centrifuge away the dead cells and fix what's left and get good results. In more standard patient samples, we've seen that we can go down to 40% viability and still get a technically pass filter library. I will always say the more viable your cells are, the better, but it does seem like we can that it is a little bit lenient to having some samples with lower cell viability, and you still can get some good results.
Fantastic. And then, so another question that's come in is, thank you for the amazing presentation. Could you please introduce me to a reference of how antibody tagged DNA could be used to detect the place of its target? I'll open that up to either Petter or Jessica.
I can mention that there is a publication on the way out, which is accepted and it's in press, which would detail the molecular pixelation technology and how. Because we didn't have time to go into the nitty-gritty details of how molecular pixelation actually works here. And so for anyone interested in reading more, there's a paper in press. It's currently available as a preprint, but the final paper is coming out in a few weeks.
Fantastic. We also have a question asking, could you please elaborate on how the method works more stepwise? I think perhaps this is not the space for us to answer that, but we do currently have a InFocus live online with Pixelgen detailing the more stepwise and in a video and an infographic to help explain those. So please do find that InFocus online to check out that question. The next question is and is addressed to Jessica, I believe, is it possible to examine the P53 expression and if it co-localizes with CD82?
Yeah, so right now for the MPX data, it is cell surface protein. So things that would be there on the cell surface. And maybe somebody can correct me if I'm wrong, but I think the P53 is going to be inside the cell. So it's going to be a little bit harder to do that today, but I don't know if somebody from Pixelgen is on the line and can maybe think a little bit about when it's going to be possible for us to do proteins that are not on the cell surface.
Yeah, I believe for now that it's purely cell surface.
Yes, I know.
But yeah, I assume
but it's an interesting idea.
Moving forward, yeah. And then the oh, sorry.
No, I was just going to say, now that CD82 has been revealed through the molecular pixelation as an interesting protein, it would be feasible for you, Jessica, to look with imaging, for example, at P53 and in relation to CD82. And so I think this illustrates how molecular pixelation will generate new hypotheses for you to then follow up in other methods.
Fantastic. And then another question that we've got through is, how is molecular pixelation better suited than in situ studies? Is it more suited for liquid biopsies and or suspension cells?
Yeah, so what we've talked about here is suspension analyses. There's no reason why molecular pixelation in the future would not be feasible also in a tissue context, solid tissue. But for now, the methodology is available for cells in suspension, and that's similar to cytometry methods, etc. And so that would either allow yes, so that's the main reason here. And then the other thing is, if you want to compare it to in situ methods, then this is higher dimensionality. You'd be looking at more proteins per cell than your typical sort of state-of-the-art spatial methods.
Fantastic. And then Jessica, as you've conducted orthogonal validation assays, so standard immunofluorescence assays, could you share your experience with the validation process? Were there any specific challenges you encountered? And do you have any recommendations based on your findings?
Yes, so all of the antibodies for MPX were made in mouse, and actually finding antibodies that were validated for immunofluorescence has not been easy. However, we did get conjugated primary antibodies, and those are seeming like they're working very well. You have to test every one that you get and optimize for your cells and your conditions, but it's the conjugated antibodies have been working quite well.
Fantastic. And then are there any particularly intriguing or unexpected results that you've obtained using this assay? I'll address that first to you, Jessica, but then we'll come over to you, Petter, as well.
Yeah, I mean, I think that for me, the most interesting thing that we pulled out of this was actually the colocalization and the polarization. I think that being able to see these data in those those modalities of the data have really kind of pushed us further in where it's pushed us beyond what we were expecting, that we would be able to answer some of the questions that we've asked when we've done the molecular pixelation in this drug screen context.
Fantastic.
Yeah,
and Petter,
I mean,
for you?
I agree. I mean, you know, we're finding all kinds of patterns of proteins that are co-localized in ways that we had no clue about in the past, or that potentially there might have been, some of those might have been reported from, you know, 30-year-old microscopy studies, but have been forgotten, and now we're sort of re-envisaging that, and so it's this really intriguing new dimensionality of the data that really offers new insights into how cells are regulated. And in my context of immune cells, it's really fascinating, because everything we're finding with respect to protein polarization, protein-protein interactions is basically novel, or offers new hypotheses. So it's fascinating, actually.
Fantastic. Well, thank you very much, Petter and Jessica, for answering our audience's questions today. Before we wrap up, do you have any last comments that you'd like to add to conclude your presentation?
I mean, I just want to encourage everyone to give it a try and see what you find in your settings. Every researcher has a different experimental system, but I am sure that this methodology has something to offer for anyone who's interested in the analysis of proteins in individual cells.
I can just add that you don't need to buy expensive instruments. If you have a basic molecular biology lab, you can make these libraries and then send them off to any standard core sequencing facility and get your results.
Fantastic. Well, I'd like to thank you once again, our wonderful presenters, Petter and Jessica, as well as you, our listeners, for your time and questions. Don't forget to visit us at www.biotechniques.com for more webinars and to check out our In Focus with Pixelgen on molecular pixelation. Thank you again for attending, and goodbye.